So what was the idea behind optogenetics? So when you look at your cells or any kind of samples, biological samples under your microscope, you see, and you do that in live, you see beautiful movies, like for example this one here, where you see protein activities, for example, that shows very nice patterns in time and space within the cell or within the tissue, if you look at a larger scale. And that's something that the fluorescence microscopy was offering, and that has revolutionized cell biology. But actually, when you look at those movies, something that you would like to do is really to move a bit further from just seeing, observing your sample. And you would really like to manipulate that as well, you know. Oh, if I had the possibility to really change this pattern and activate my protein, for example, here in the cell. And this is what I call cell biology 2.0. And the idea is that now we can do that actually. So using light, and I will explain you how it works, but you can, under your computer, click on a cell and say that you want the protein to be active here. And then you do that, and the cell is going to activate its signaling pathway locally using light. So I'm going to explain you how that works and which are the different toolkits that you can use for that and what are the applications you can think of. So just to recall what is optogenetics coming from, actually at the beginning it was used in neurobiology. And this is one of the, Daysroth lab was the first one to invent that term and that technique. And what you see in this experiment is a mouse that has a little LED implanted in the brain, a blue LED. So this one is wireless on the right. And what you see here is that actually here the blue light is used to control the running behavior of the mouse. And as soon as you turn on light, you will see the little mouse start to run. And as soon as you stop light, it stops. So it's a pretty powerful control. So this is what people developed in neurobiology. But actually, now we can do the same with cells, actually single cells. And this is one of the first papers that did that in Klaus Hahn lab here. And the idea is that here they use a little laser spot that you see here. So you shine it in the front of the cell. And then basically you remote control the migration of a cell. So it's a bit like the mouse, but not at the cellular level. So the idea is trying to explain you how that works actually. So just before, coming back to this neuronal optogenetics, what was the idea behind that? Actually, the idea was to use some biological systems that are photosensitive. And for neurobiology, actually the idea was to use an alga, so it's a single cell alga that has two flagella here, that has actually properties to sense light and react to it. And actually when you shine light on a dish here where you have this alga, actually it moves against light. It don't really like light, it likes dark spots, and it just do this phototaxis, but in the other way, like going away from the light. So how this simple single cell organism do that? It actually has an A spot. If you look, it's like an assembly of receptors on one side of the cell. So these are transmembrane receptors that are light gated, which means that when you shine light, blue light on those receptors, actually the channel opens and lets the ion pass through it, which then leads to the signal like you have in neurons, actually, the same idea of ion-gated channel, but here it's light-gated channel. So the whole idea of optogenetics at the beginning was to actually reuse the gene that is encoding for these light-gated receptors, which is called channel rhodopsin, and to put that into the transgenic mouse and under the expression of specific neurons. So just the subset of neurons that are responsible for a given behavior will express those light-gated channels. And then using light, you just shine light on the mouse and it will basically activate the neurons by this light-gating mechanism. So that's the idea for neuronal optogenetics. That was the origin. But now, actually, optogenetics has been also extended to non-neuronal application, and this is what I'm going to talk about today. And the idea is still the same, is to reuse actually some proteins that are photosensitive, that will react to light, and then engineer those proteins to make actions on your biological system. So the general idea is that you have a protein that you can encode, so genetically encoded, and then it will do some change of conformation and become reactive. And you are going to use this reactivity to tune the biological effect. So one example that I'm not going to discuss, but it's actually expanding a lot right now, is, for example, to control gene expression. So just doing this, using this system, you can engineer a system to control the expression of genes. And you know, all this clonal analysis that people do in tissue, now you can do that, but with your own spatiotemporal properties. So you can select, pick up a cell here, shine light on that cell, and decide which kind of time course of gene expression you would like to have, and then using a system that is photosensitive, you will achieve this expression. So I'm mostly going to speak about signaling pathway and signal transition, and I will just quickly present now the different toolkits. So it's quite a mature field now, and there are several modalities to control the activity of proteins inside cell, and I'm just going to review them now. So just the tools themselves on the molecular aspects, you have different modalities. So it depends actually, I decided to classify them as how many proteins are involved for that. So you have this one protein system, so it's like intramolecular, where the idea here is to use a light-gated domain here that will change conformation when you shine light. So when you shine light, you're going to change the conformation, and that's why you can, for example, open a binding site that is hidden before, and then the protein becomes reactive because it can interact with partner. I'm going to discuss a lot because this is a very versatile approach, the one that is involving two proteins. So it's intermolecular interaction that you control. And the idea here is to have two proteins that are going to form dimer when you shine light. You are going to form a dimer. And this way you can actually bring the green protein with the red protein. This also can be done with homodimerization. So some of these proteins have the tendency to form homodimers when you shine light. And this can be of use also. And I will also discuss quickly this control of a large assembly of proteins, where the idea is to form clusters of proteins or aggregates of proteins using light. Yeah. So to give you some example, I will start first with this intramolecular system. So actually this is the system that was used in the first movie I showed, where with a laser spot you remotely control the migration of a cell. And what the idea here is to use actually a fusion, which is shown here, where you use the RAC1. So RAC1 is a small RhoGTPase, which is involved in cell migration. It's a RhoGTPase, which is either inactive or active, depending on its GTP or GDP bound state. And in the active state, it recruits all the active machinery that promote the cell protrusion, and that makes the cell move forward when it's crawling on the substrate. And the idea is to engineer a protein that is constitutively active, so it's always active, but the binding site to effector is hidden by a love domain, so which is this domain that changes conformation upon light. So then when in the dark, no effectors can bind to the protein, but when you shine light, you will open these loops in the love domain that will let the effector bind to it. So this is one example here where you see a mammalian cell that is just a single mammalian cell that is on a cover slip, and that is expressing this construct here. And here, the bright spot that you see is a laser that you shine. So you see the reflection on the cover slip of the laser, actually, that lets you see where you shine light. And you see that every places you shine light, basically, you make the cell basically activate all the RAC1 signaling pathway that ultimately leads to this actin polymerization and branching and protrusion. So they call it like the cell painting. You can paint the border of the cell as you like by just using this laser here. And this has been used actually to control cell migration. And one nice example that I like actually is the ability to control one cell, it's a neutrophil, in a living zebrafish embryo. And what you do here, so there was a guy like just using a laser to make the cell move forward as he wanted to. And he spent a lot of time actually because maybe the experiment runs for hours here. And he actually spelled the letter of RAC just by using migration of this neutrophil in the living embryo of a zebrafish here. So you see how tightly you can control the migration here. So another application of this just intermolecular construct. So instead of doing this kind of cage approach where you cage the binding site, you can cage, for example, a degradation tag. So it's a tag that will send the protein to degradation, but if it's hidden, the protein will not be degraded. So the same way you can actually present, when you shine light, this tag that will send the protein to degradation. So it's a way to control degradation of protein. And here are some, I just show you the movie. It was done on yeast cells here for the degradation of the red fluorescent protein just as a proof of concept. And what you can see, for example, if you shine a pattern on the plate here, basically, and this is a fluorescence image, you can pattern that in fluorescence within the cell just by degrading the protein, since it's linearly responding to the intensity of light. So you can really do grayscale degradation control of protein. So very quickly, another approach for that I will, oops, sorry, it's not here. Let me move, no, it's not here, sorry. Another approach that was used based on the same idea is instead of a degradation tag here, you can put an NLS, a sequence for nuclear localization signal, and you can play with the nuclear to cytoplasmic exchange by light. You can send the protein to the nucleus or send it to the cytoplasm with exactly the same idea. So something that I will discuss a bit later is actually this idea of now dimerization. So you have two proteins now. And actually the main use of this approach here is to put one of the proteins here, fused to the photosensitive partner here, to put this tag that will target a specific compartment within the cell. So you will have an anchor that will be light sensitive, light activated, and then you have the protein of interest that you can fuse to the other partner. And then when you will shine light, basically you're going to send this protein to the compartment that you want. And that's the way you can manipulate the localization of protein within cell. And here I give an example in yeast here, where the anchor here can be of different variety. So you have like a tag that will target the plasma membrane. So you can send, like you're initially in the cytoplasm, and then you shine light, and almost immediately the protein is sent. So this reporter, which is just a fluorescent protein in that case, is sent to the plasma membrane. But then, of course, you can target the butt neck, the many, like the nucleus, the like almost all compartments in the cell where you have known anchor for that specific compartment. So this is very useful, as you will see, because it offers a way to be to basically control almost any kind of signaling pathway. So on the dimerization, so something that is also using two proteins, but now to make homodimers. And here is a nice application of that, is using these growth factor receptors. So if you know how growth factors work, actually, they bind ligands, and once they bind a ligand, they form dimers. And once they form dimers, there's all these phosphorylation cascades that take place. And here the idea was to use these FGFR receptors, cut the extracellular domain here, to just keep the intracellular domain, and then fuse it to a light sensitive protein, which is called Cry2. It's coming from plants, actually. And this Cry2 has a tendency to form dimers, homodimers, when you shine light. And actually, using that, when you shine light, you are going to make this intracellular domain dimerizes. And that way, you can activate all the growth factors signaling transduction machinery that is behind. So what you see here is actually fibroblasts that are not light sensitive at all, normally, but that express this construct here. And actually, you make this fibroblast being able to do phototaxis by just having this system. Like in this experiment here, blue light is shining in this circle, and you see that all the fibroblasts wants to migrate in that place here. Okay? Because actually the light is like for them, they receive growth factor, so they're pretty happy to be here. And this is why they all migrate here, and they do this phototaxis here in these cells. So last modality that I present is this formation of clusters here. So there's basically two ways to do it. So either using multimeric protein. So here again, the idea is that you have a binding dimerization between one protein and another one. And if you put one on the partner on a multimeric protein that has multiple sites, you are going to form this large assembly. And as the protein has also a tendency to aggregate, you will form these large clusters. And that's a way actually to basically, from a diffuse localization in the cytoplasm, form localized puncta within the cell. And you see here, for example, like 3 cells, and if you can shine light on cells, and then select the cells you want, or it can be even subcellular. You see, you can form these clusters. And actually, all the tools I'm presenting here are reversible, so which means that when you stop shining light, everything goes back to the original state. So actually, I present that because this is a quite useful techniques, because it allows you actually to do one thing, which is fun, to sequester almost immediately a protein. Imagine that you have a protein doing a job in a cell, then you shine light, and immediately the protein is sequestered in these clusters and cannot do its job. So it's like, you know, doing a knockout or deactivation of a protein, but instantaneously. And this is actually one example here that I show you with like different proteins here. What you have here is, for example, a GEF that activates a RAC protein here. And as soon as you shine light, for example, you will see this protein will be trapped in clusters, and the cell retracts and contracts. You can do the same for the CDC42 itself. And it works for almost any proteins, actually. And the other application of that, actually, is to actually also trap proteins, for example, that has a GFP expression domain. So if you put a nanobody against GFP on your oligomerization technique, basically you can trap any GFP tag protein. And I think here is a movie where you, oops, no, the movie is not here, sorry. Yeah, where it's a bit hard to see, but it was tubulin that is trapped. And basically you impair the mitosis, so the cell cannot divide anymore because the tubulin is trapped into clusters. And cells are stopped during division, what you can see here in this arrow here. So actually, there is a more simple way to do these clusters. Now with just one protein, actually it's a modified version of this Cry2, cryptochrome 2, coming from plants, that has been engineered to do this oligomerization state. So now you can just have just a tag, like a GFP tag, but that is clustering. And this is what you can see in this movie here, this induced clustering with just one tag. So it's pretty efficient, actually, it works very well. And again, you can do the same application as before. And here is another application where it's actually used to do this bait and prey experiment. So the idea is if you want to know if two proteins interact inside cell, you are going to cluster one of them and see whether or not the other one clusters. So here, for example, you have a case where the two proteins interact. So when you cluster the red protein here, which is fused to this Cry2 oligomerization system, you see that the green one is also clustering. So you know that they are interacting. Whereas in that case here, you make cluster, but the green protein doesn't make cluster, so they don't interact. So it's a very nice way to assess interaction. Okay, so to conclude on that kind of general overview of the tool, you have basically two kind of light-activated systems. The one that are in the blue, which are actually the love domain, so the one that changes conformation upon light, and the cryptochromes coming from plants, actually. And you also have red activation system, mostly the phytochromes, one that are coming from microorganisms, bacteria. And the idea is that, you know, one of the problems also with optogenetics is that you need light to activate the system, so they are light sensitive, which means that if you want to combine activation and observation with fluorescent protein, then you cannot observe in the channel where you are going to activate, of course. So there is then a problem of jamming of wavelength. And for example, when you work with blue system, you cannot image CFP, GFP, YFP because you are going to activate. So you are left with red or far red protein. The other way around, if you work with red photoactivation protein, you cannot use red fluorescent protein or infrared one. But actually, quite luckily, there is now like a large panel of fluorescent proteins, even up to the very long wavelengths. So you have, in general, two channels in addition to the optogenetic one that lets you see what you do, and one to see the effect on the biological system. So this is usually what the people do here. So there's a nice review here that you can read about those systems. So they're always quite sensitive and quite efficient, which means that all the activation takes place in a few seconds, and they are reversible also pretty quickly, like in a couple of seconds for some of them, a couple of minutes, then you go back to the original state. Yeah. So in terms of optical device, so which kind of microscope do you need to do that? Actually, there is plenty of options that are available. So if you just want to do temporal control, you know, shine a pulse, activate for 2 minutes, and then stop it. You can use any microscope setup, even use the white lights, the LED, or the fluorescent imaging lamp to basically do this temporal control. And you also have some cheap solutions if you want to work with plates, for example, 6-well plates or other. You have solutions with Arduinos, for example, that explain how to put some LEDs and give some patterns of light over time that you want to achieve. If you want to do some subcellular or select some cells, like to do spatial patterning, it's a bit more complex, but also, again, you have many solutions. What you need at the end is a microscopy setup that will allow pattern elimination. And in general, you can the main system that has been used for that are the FRAP system. So, for example, where you have a focalized laser that can move around the sample, so you can activate just at the place of where the laser is focalized. Or actually, something that is expanding now is the use of DMD, so which are micro mirrors technology, actually the same that you have in the video projector here. So in the video projector, you have a chip of micromirrors that shine lights that flip very quickly. They're on and off. And based on this on and off modulation of the frequency, you can shine grayscale pattern. And if you couple that, if you put a video projector on the microscope, you can couple that to the microscope, then you can project a movie on the cell, and that way you can activate with like almost any kind of pattern on the cell. So I will now comment on something that we worked quite a lot. So based on these two proteins that form dimers, and the idea was to use this system to manipulate the distribution of protein in the cell. So now I will describe you a bit more in detail, and that will let you understand all the biophysics, how these optogenetic systems work. Yeah. So actually, what I'm going to present now is the use of this, again, two systems where you have one photosensitive protein. In that case, we used the Cry2, so the cryptochrome 2 coming from plants. And that is actually normally in the plants used for the circadian rhythm. It's like it's reacts to blue light, and it's actually used in plants for functions. But the idea is that if you put that into mammals, then it will not interact with any kind of signaling pathway in the cell. It's completely orthogonal. And then you can use the property of this dimerization to design a new way to control cell activity. So here we use this Cry2 protein, and its partner is called CIBN or CIB. And in the movie I'm going to show you, this partner is anchored to the plasma membrane with a specific polybasic tag. And this Cry2 is fused to M-cherry protein, so here it's just with fluorescent protein to understand what's happened. And when you shine light, blue light on Cry2, it will be change conformation, become reactive, and will bind to its partner. So basically, you are going to send this protein to the plasma membrane. And this is reversible. It takes about 3 minutes for the complex to dissociate in the dark. And that was actually published in 2010 here. So in the movie that I'm going to show you, actually, most of the movies here are done in turf microscopy. So if you don't know what is turf, the idea is that, so here is a side view of a cell on a cover slip here. And the idea is to excite from the bottom with an angle so that the light is bouncing on the cover slip. But there is a little field, which we call the evanescent field, that is propagating above, and which is just enough to eliminate the ventral side, the ventral membrane of the cell. So that's a way to just see the ventral membrane of the cell that is in contact with the cover slip without having the signal that is coming from all the cell. So this is pretty convenient because if you want to see something localized on the plasma membrane, then what you see is just some signal that appears because it's coming to the plasma membrane. So here, for example, you have two cells that you see in turf. So you don't see the nucleus because, of course, you see the plasma membrane. You have what we are looking at actually is this Cry2 M-cherry fusion here. And you have a bit of signal because actually there are some molecules that are bouncing randomly onto the membrane. So you have a bit of signal. But when you shine light on a specific position, this is what you're going to see in a second, the movie is looping, then you start to see patch of recruitment on the plasma membrane. So it's really protein that you activate. Here it's activated by a focalized laser. So in the cone of light, you activate the protein, they diffuse, and then they come and bind to the plasma membrane above and below, and you see this population that is coming below on the cell here. So that's and you see that if you stop illuminating, basically the protein dissociates and go back to the cytoplasm here. So if you detail a bit more about the numbers of this membrane localization, actually when you shine light, it takes about 3 seconds for the protein to diffuse in the cytosol and get into the plasma membrane. And then because you illuminate in the cytoplasm, the protein diffuses a bit laterally before reaching the membrane. So you have, like, even if you just illuminate on the one very small point, you still have something that is a bit spread out on the membrane because of this lateral diffusion. So this is what you see here, for example, in that experiment. What we do is that we shine light on a small point here, and we look, and we just play with the exposure time. So how long is the flash of light? So for a small flash, you have a small recruitment, and as the time here gets longer, you have a bigger amount of protein that is recruited. But you see that you have this kind of lateral extent, which is about a couple of microns here. So then the complex on the membrane diffuses on the membrane because of like membrane diffusion with a diffusion coefficient of 0.1 micron square per second. There is a square that is missing here. And then dissociate in about 3 minutes here. So actually, you can modulate the system by, for example, playing with the time of exposure, but also with something that is more useful when you do time-lapse imaging on microscope is to play with the frequency of pulses. So when you do flash of light. In that case, for example, if you do like one flash every second, so this is the maximal recruitment, you are going to deplete all the cytoplasm and put it on the plasma membrane. And then if you decrease, you do one flash over 20 seconds or one flash every 60 seconds, then you can modulate the amount of protein you want on the plasma membrane like that. So this is to give you some order of magnitude of the numbers for that. And of course, this is related to this dissociation time here. So yes, this I will skip. So now if you want to control a steady state in space, so imagine that you want to apply a perturbation locally in the cell and then maintain that over time because you want to see what's the effect on the cell when it's integrated over time. So actually, because the protein is diffusing and dissociating, if you constantly eliminate, you are going to get a steady state, which means that you recruit constantly, protein diffuse, dissociate, and you recruit them again and again. And that way you can maintain what is called a steady state, because it's constantly dissociating and reassociating. And from physics, it is well known that such a system, the solution of it actually is an exponential distribution at steady state. It's like with morphogen gradients, for example, when you have a localized source, diffusion and degradation, you form an exponential gradient, decaying gradient. And the length scale of that gradient is actually related to the diffusion coefficient, in that case of the complex on the membrane, and the lifetime of the complex. And in that case, it's about 5 microns, if you just take those numbers. Experimentally, it's a bit more, it's 10 microns, because of this first step of lateral diffusion. But so this is one example here where you see in turf again, in false color, the formation of an exponential gradient along one side. So we activate here on the left part of the cell, and you see that the exponential gradient is quite stable. So you can maintain that over, it's almost an hour here. You maintain that gradient here. And actually, this is a bit the same principle that the cell itself is using to maintain asymmetrical distribution of protein in the cell. So this number is important because this is somehow the resolution that you have with the system. So you cannot activate something smaller than that because you're limited by diffusion. So this is an important number that tells you the resolution you have on the system. So that's kind of the summary of for that system, the important number. So this length scale. So the on time is very quick. So in a few seconds you activate, and the off time is a bit longer. And knowing this number, you can basically know how you can engineer, how you can manipulate protein inside cell. So for example, here again, we move the cell, make a gradient from the left, from the right to the left, and then make a gradient from both sides. Actually, you can do whatever distribution of protein, given that it's not sharper than these 10 microns, and that you don't move the whole gradient faster than these 3 minutes time scales here. So some people are trying to improve those numbers because actually some of the patterns that you see within cells are smaller than that. Some cells are even smaller. So if you work with the Drosophila cell, for example, they are 10 microns. So they are too small, and you cannot do something local. So it's important to improve that. And actually, the two ways to improve the resolution is either to lower the diffusion, so to have something that diffuses less, or to have something that has a lifetime as a complex shorter. Because if it's shorter, then it will decay before it will move away from the places where it was activated. So that's the way. So there is another way, which is actually using this phytochrome B system. So this is another dimerization system, exactly the same one, but using phytochrome, which is very nice because actually you can use a given wavelength. So it's at 650 nanometers, you activate the dimerization, and with 750 light, you inactivate it. So you can actually, for example, target a specific place where you do activation, and then you shine inactivating light everywhere else. And if you do that, then you are going to keep a very sharp distribution, because as soon as the protein moves out of the activating domain, it will be deactivated by the inactivating light. But actually, this is a pretty hard system to work. You really need to tighten the expression level of both proteins very tightly. We spend a lot of time in the lab trying to make it work, and it doesn't work at all. So I just mentioned it, but it's a pretty tough system to work with. What I think is going to be the future is more, as for fluorescent protein, people make new generations that are more bright, more photosensitive. Some people also engineer new dimerization systems that are more sensitive, more efficient, have a larger dynamic range. And one of them, which is very nice, is called the eyelid system that was published recently. It's the same idea as the Cry2 SIB system I presented. instead that it's inverted. The photosensitive protein is on the plasma membrane, which is a bit better because then when you activate, it doesn't diffuse in the cytosol before binding to the plasma membrane. And also, it has a lifetime as a complex shorter, like less than 30 seconds. So because of that, actually, the spatial resolution is about 2 microns, which is something very nice. With 2 microns, you can do a lot of things. So for example, in that case, by the same idea of recruiting to the plasma membrane, as I presented before, they could spell the letter I, L, I, D in the cell. So you do like a really like a sharp pattern as manipulation of protein in the cell. So now in the time that is remaining, I will just show you what you can learn, which is new, actually, because this is a very nice technique, but is there something really new you can learn from that about biology? Because this is what you would like to know at the end. And I will just pinpoint a couple of ideas, which is about signal transduction and what is new. So one example that was published from another group from the US, which is very nice, that used actually this phytochrome system, the phytochrome B PIF system, which is activated and inactivity, the one that doesn't work. But actually, it's a pretty good lab, and they succeed in really making this tool work by using lentiviruses, so stable expression of cell, faxing them to get exactly the good level of the expression, and they worked a lot on that, and actually they're pretty good with this system. They're the only one that knows how to make the system work. So what they did actually was to use this dimerization system. One is on the plasma membrane, again, as before. The other one is in the cytosol. And the idea is that this partner now is fused to this, what they call optosource. So it's a source, it's a GEF domain that is activating the MAP kinases pathway. So this ERK, RAF, RAS, MEK, ERK, all this kinases pathway that is successive phosphorylation up to the double phosphorylation of ERK. And when ERK is phosphorylated, what it does is that it's shuttled from the cytoplasm to the nucleus. So basically, to test the system, what they do, they take cell lines. Here it's a fibroblast in that case. Before it's in the cytosol, this optosource, you shine light, it goes to the plasma membrane. And if you look at a fusion of ERK with a blue fluorescent protein here, what you see is that you have a localization from the cytoplasm to the nucleus. So you activate really the signaling pathway that way. Because this source actually is inactive in the cytosol, it's only active when it's on the plasma membrane. There are many, many reactions in the cell that work like that, inactive in the cytosol, but when you concentrate it on the plasma membrane, where all the proteins are, then you trigger the signal on. So here you see, for example, like now, it's like an assembly of cell where they shine light with actually here random levels of light. I will explain you why in a second they do that. And what you see is that then they can measure both two things at the same time. One, which is actually the level of membrane recruitment. So this is the input that they have on the signaling transduction machinery. And then they can look at the shuttling of ERK into the nucleus. So you really have a quantitative way to measure the input and the output at the same time of a whole signaling machinery here. And using that, actually, they did very nice quantitative work on that you could not have accessed before. And one that I like is actually, if you just now pick up one cell, and as you shine different level of light, you can basically sample in this input-output map the response of the cell. So each point here is a given level of optogenetic activation and a given level of cell response. And so you can draw this kind of dose response curve, but at the single cell level. And this is very accurate. And something that you can learn from that is that when you do that over many cells, actually, so people already did that before, but using population measurement. You know, you can do that on a fax. If you use a fax, you just sample the whole cell, how they respond to different levels. And you end up in general with this kind of graph here, where you have a huge variability here between the input and the output. And actually, when you look at this graph here, basically, you don't know where this variability is coming from, if it's single cell or just the population. But if it's single cell, you would say that, okay, given this variability, I would say that the cell can just have two levels, you know, like a high level of nuclear ERK and a low level. Because if you look at how many states you can encode with that, basically there are just two states, a low state and a high state. And you'd think about this system as just being on or off. You know, either the MAP kinases pathway is on or it's off. That's the only way the cell can do that. But now if you do that at the single cell level, actually what the people has realized is that these input-output relationships are much more precise than this population measurement here. And what you say is that every cell has a different input-output relationship. Okay, there is a large cell-to-cell variability, but within a cell it's very accurate, which means that actually every cell can encode much more than just one level, low or high. So it's not digital at all. There are much more information that the cell can encode with that. And that's something that you can realize by having this ability to really do this optogenetic activation within cell. And now I will speak a bit more about what we do ourselves, which is a bit in the same spirit, so trying to do like quantitative measurement of input-output relationship in signaling transduction. And we work mainly with RhoGTPases. So I talked already a lot about it, but here to give you more detail, the way RhoGTPases work is that they shuttle from the cytoplasm to the plasma membrane. And when they are on the plasma membrane, they are activated by GEF, guanine exchange factors, so which means that does the exchange between GTP and GDP. And in the active form, bound to the plasma membrane, they recruit tons of effectors. And they are deactivated with gap, so there is like all this cycle of activation, deactivation that takes place. And the idea of optogenetics was to actually use this GEF, this activator here, but you truncate them. So you just keep, so that these are multimeric proteins having many domains. So you remove all the domains that are here, for example, to localize at a good place, at a good time, the protein on the plasma membrane. You just keep the catalytic domain, so the one that is able to activate the protein. And then this catalytic domain, when it's truncated, it's in the cytosol, and it never goes to the plasma membrane, and it's inactive. And then you fuse this catalytic domain to the protein that you are going to recruit to the plasma membrane, and that's why you can rescue actually this membrane localization that is used to activate the RhoGTPases here. So you can do that for the main, all the RhoGTPases, the canonical one, so RAC1, I discussed it, CDC42, which is mainly involved in Philippodia formation, and RhoA, which is used for cell contractility, like contraction of cell and cell, and also during cell division for the separation of the two daughter cells. So again, the idea is that on the same kind of graphics here, now you put an active protein, and that you are going to be able to rescue this membrane localization. One advantage is that actually you are going to activate only the endogenous pool of RhoGTPases, because you don't overexpress the RhoGTPases. Compared to these RAC1 techniques I presented at the very beginning, here you don't overexpress the RhoGTPases itself here. So this is one example here with CDC42. So actually CDC42 is making Philippodia, but it's also like involved in many other processes since it has a lot of crosstalks. And what you see here is the fibroblast where you activate this CDC42 using this Cry2 CDC42 OptoGEF system. So what you see here is the turf movie, so you see exactly which kind of gradient you form within the cell. And here you see what the cell is doing. So it's forming this Philippodia, but also this huge ruffle and this huge vesicle as well. So there's plenty of pathways that are activated that actually ultimately lead again here to the migration of the cell. So initially the cell was not migrating, and basically you force it to migrate in the direction of the activation here. And you see this activity here. So that was already done with other techniques. So something that we actually developed in the lab that was not yet done was using the same idea to play with cell contractility. So now if you put the GEF for RhoA, which is a protein involved in cell contraction, and also in cell retraction during migration, what you see here, so you have a cell that is almost round at the beginning, and then you are going to recruit in that box here, the activator. And so what you see is that the cell in the box retracts. So this is something we expect, because you activate RhoA locally, so the cell retracts locally. What we were surprised actually is that if you look carefully, actually on the other side of the cell, exactly at the same time, the cell moves forward. So it's like you press something that moves in the other direction. And that was surprising because it's really exactly concomitant, taking place at the same time. So based on this observation, actually what we first did was to really look about the RhoA signaling pathway more in terms of input-output relationship. And we also studied the way the coordination between the back and the front of the cell is happening in that case here. So just to give you a very quick idea about that, so in terms of this input-output relationship, the idea is that you control this complex signaling pathway of RhoA, with the optogenetic activator, and then you look at something else downstream of the activation. So RhoA basically is doing two main things. One is promoting the actin polymerization through formins that actually nucleates actin. And also it activates some kinases that actually play with myosin to activate myosin, to do the real contractility. So it does two things. First, make actin, and then also activate myosin for contractility. So the first thing that we looked at was actually the link between RhoA and actin polymerization to see how in space and time the activation of RhoA was acting on actin. So the way we did it actually to analyze that was to make a kind of quantitative person chase approach. So it's the same idea of having an input and an output that you measure at the same time, but now you have space in addition. So it's just not only time, but also space. So the idea is that you sign a puff of activation with the optogenetics. So this is the recruitment after, and this is the differential. So after minus before, the image, and you see this puff of recruitment. And if you unwrap that, so if you take a slice of that and you look at it over time, you see this kind of puff of activation, so which is local in time and local in space. And the idea is that then you can look at the same time as a reporter of actin polymerization, for example. So in that case, it's F-actin, so life act, to see the branch, the polymerized actin. And you also see a puff or response of the cell, which is polymerizing actin. And the idea is then to relate this to that in both space and time. And actually, in space, it's quite linear, which means that you cannot see it really here, but if you look at the relationship between the local activity of RhoA and the actin polymerization, it's just a proportional factor. So it seems to be very linear in space, and it is also in time. I go fast because this is a bit complicated, but the idea is that if you look in time at the total signal, so here are the pulse of activation and the relaxation. And the idea is that you can measure that. So this is the experimental input. So this is the recruitment on the plasma membrane that you measure from the microscope. And then you can feed that in a little model where you say that the actin is either monomeric or filamentous on the cortex. And that you have some exchange between both of them. And actually with this simple model, you can feed that curve here in this model and see what's the best predicted curve you can get. And actually the two numbers is that at which rate actin is polymerizing and at which rate it deactivated. And these are the two rates here. And you see that with the activation, actually you constrain a lot one of this rate, which is the deactivation time, and which tells you that it takes about 3 minutes for actin to depolymerize. But it's not only depolymerization, because depolymerization is much more quick. Here what you really measure is the whole signaling transduction. So you see the whole time you need to deactivate the whole pathway. Because when you activate molecular species, you activate them, and you need to deactivate them. So it's like all the deactivation of the intermediate state that you measure at the same time here. So that's one example. So the idea is that yes, you really measure with the perturbation the whole dynamics of the pathway that you disequilibrate toward a given state. And also with the relaxation, you see how that goes in the other direction. And that's the kind of numbers you measure here. And something that we learned from that is that actually it was also very linear. We expected to see something highly non-linear, which means that as you increase the amount of activation, the amount of polymerization will just explode or increase a lot, because actually many of these steps here are catalytic, meaning that one protein is able to activate many of other ones. But actually it turned out at the end that it's very linear. It's a very simple linear system here. So just a last example of what we did with that was, as I told you, the cell is moving in the other direction when you locally contract it, and we were wondering what was happening. There was several options, So it's all about what is the front and the back coordination in cell migration. It's a hot topic. There are many options for that, and maybe Paolo will speak a bit about it. But so to give you an idea of the different models, so one of them is based on membrane tension. The idea is that if the tension is high in the back of the cell, then through the cortex and the membrane itself, there is some mechanism that will coordinate the front of the cell based on membrane tension. So other people propose that it's actually based on the pressure of the internal cytoplasm. It's like you all played with a glove full of water. When you press it, it makes the pressure goes along the other part of the gloves. So that's based on the basic physical mechanism. Some other actually models are completely different. They are based on reaction diffusion. The idea for them is that to have crosstalks between RhoGTPases themselves. And the idea is that RAC1 and RhoA has mutual exclusive inhibition. So when you have high RhoA, in general, you have low RAC1. And if you have high RAC1, you have low RhoA because they inhibit each other. So the idea is that through diffusion and reaction, you are going to activate RhoA, and then it will deactivate RAC1 locally and upregulate it further apart. So this requires diffusion of molecule in the cell. And there are plenty of data. So the other model actually based more on the actomyosin contractility that Mathieu Coppey, the collaborator of Paulo, worked a lot, where the idea there is more based on the retrograde flux of actin. So the actin is coupled with myosin, and then it will contract, and you have all these actomyosin flux that are known. And having this flux, basically, you can move the protein and relocalize them somewhere, and delocalize them somewhere else. So that's also another mechanism. So there were all these mechanisms possible, and actually we use optogenetics to dissect what could be the good mechanism for that, and this is really a nice tool for that, because you have the option to really do the thing, activate it. So you have before, you see your cell before, and then you do the activation and see what happens. So it's a pretty powerful to really see to dissect the causes and the consequences in these things. And actually what we saw is that to summarize the story, is that when you activate RhoA, so these are movies actually, I don't know if you can see it, but they are movies. When you activate locally, of course you see this local recruitment of actin, as I presented before. And because you have more actin, actually you have more myosin too, also that recruits it here, because myosin is targeted to actin. And actually the idea that we see is quite simple. This coordination, what we think is happening is that as you localize actin and myosin in the back of the cell, basically you are just going to deplete it from the front, just based on a simple mass balance. Okay, if you just put more there, then you remove the other from the other side. So this is what you see in this chemiograph. Like as you look at actin, you recruit actin here, and actually it's getting depleted in the front. And the same is happening with myosin. And this is what we think is happening in that case. So it's really a mass balance that is coupling the front and the back. So very simple system again, but which is interesting for several properties. I will go fast. These are the models just to show, but it's quite easy. And that if you localize in one side of the cell more to the cytoplasm, then you deplete it away. But you understand that very simply with hands here. But the model predicts nicely that both actin and myosin will show this front depletion here. And the model that we have for that, and how we can use cell migration from the rear with RhoA, which is, I think, for example, relevant during contact inhibition of locomotion. So if you know that when two cells come into contact, they repel each other. And one of the questions is that we know that when they come into contact, it activates RhoA locally. And we think that maybe during that process, because you activate RhoA locally, then you activate RAC1 on the two other sides of the cell, and that's the way the cell can repel each other. And that could be used exactly the same mechanism as here. And so to couple that to cell migration, the idea is that you can think about the border of the cell being in a tug of war between protrusive forces that wants to make the membrane protrude and contractile forces. And you have a balance. Of course, it's dynamic, so the membrane fluctuates around or have cycle of protrusion attraction. So this is dynamic. And something that you can see to reveal this kind of tug of war, for example, if you use a drug that blocks the contractility, you can just target the kinase that is doing the contractility. What you see is actually the cell that has a given shape will spread out everywhere, like a couple of microns away here. And actually what we do with our optogenetic activating here, is actually as we recruit actin and myosin here, basically we augment the contractile forces locally, and because of the depletion, basically you decrease the contractile forces away from that position. And just using that very simple process, you can make the cell move then, because of this unbalance of forces here. And I will stop here because to keep a bit time for the questions, and just acknowledge Leo Vallon, who was a PhD student that worked a lot on these optogenetics tools that is now in Barcelona doing his postdoc, and you, of course, for your attention.